Genotyping and Microarrays by Danielle Greening. What is genotyping? Genotyping is the technology that detects small genetic differences that can lead to major changes in a phenotype. This includes both physical differences that make us unique and pathological changes underlying diseases. It has a vast range of uses across basic scientific research and medicine, and also in agriculture. Ireland is only the second country in the world to have over 1 million cattle in a valid genotyping database, according to the Irish Cattle Breeding Society Federation, ICBF. Ireland currently has the world's largest genotype database for beef cattle, with over 894,000 cattle that have been registered on this database. Genotyping is becoming increasingly popular. The assessment of an animal's genetic makeup can vary between every breed. Holsteins have a very large-scale database. This is due to the high amount of dairy industry within the country. By adding genotyping, you are also adding a reliability level to breeding on the farm. Artificial insemination companies used to depend on pedigree and generate offspring. This was to later see the dependability and the production once it had taken place. Nowadays, companies can take a newborn animal and genotype it and already have a clear picture from DNA received by the parents and from that animal on what the production in the future will become. Over 70% of semen sold now is from young bulls with no progeny, and the bull is genotyped and assessed on its DNA's capabilities and for future reference. Longer lasting, lower costing, more productive animals, then this also allows for a better progress and value to the breeding industry. What is a microarray? A microarray is a piece of laboratory equipment that is used to detect the expression of thousands of genes at the same time. DNA molecules attach to each other and to the slide and act as probes to detect all gene expression. This is also known as the transcriptome, or a set of messenger RNA or mRNA transcripts. These are expressed by a group of genes. The cost in genomic selection is usually returned to the farm very, very quickly. Essentially, genomics allows a prediction to be made for future breeding. A reference or older population is then used to compare against the new DNA. Genetic data, performance data, and ancestry data are all combined to create a genetic force. DNA testing allows for identification of what traits will be passed on to the offspring and what traits will not. This can often identify parents if unknown by farmers and allows for breed verification if also unknown. This also can identify if the individual animal is a carrier for certain diseases or for certain unwanted or undesirable genes. Genotyping also ensures that there is full traceability of each sample selected and inputted into a database. Currently in Ireland, a BVD DNA sample must be taken from every newborn calf once they are born and registered within the first 2 weeks. ICBF is currently working with the Department of Agriculture to use this as a base to also create a DNA database nationwide. Last spring, a trial was carried out very successfully on the sampling and registration of these DNA samples, both for the BVD testing requirements and also to have extra DNA within the sample to add to a database to be as a whole for nationwide use. Genomics is widely used across the agricultural sector. One of these areas being the using of DNA genotype to help better predict how well an animal will perform in the future. DNA is passed through parent cattle to offspring and therefore the main body of the decision making when breeding. These samples can be taken from blood, tissue, hair, skin, and then a DNA sample can be taken from this and used to analyse or compare with other profiles. This can then allow the breeder to make educated decisions in the selection and in relation to areas such as performance data, ancestry data, genomic data, and genetic probabilities. Cattle can be genotyped using marker sets. This can be done in sets of 11 or up to sets of 16 microsatellites. By using genotyping to see a precision picture of DNA markers, which are alleles, which are then formed into a gene to take via mutation that an individual cow has been given by their parents. Genotyping allows for genomic selection of desirable traits and the eradication of undesirable traits within breeding. Thank you for listening.